My name's Diane Bassham, and I'm a professor in the Department of Genetics Development and Cell Biology at Iowa State University. My lab is interested in questions related to environmental stress responses of plants and how plants can tolerate different environmental conditions. Our main interest is in studying autophagy, which is a pathway for the degradation of molecules in plant cells. I wanted to start just briefly by giving an overview of where this talk is heading. So in my lab, the things that we're really interested in are how do plants tolerate environmental stresses. So we're interested in, if you take a plant, you put it in conditions like drought or heat stress or cold. How does the plant tolerate that? How does it continue to survive and even grow under these conditions which are really not optimal for its growth and survival? And so that's kind of the big picture of what we're interested in. But I wanted to start by actually telling you why we're doing this research in Iowa. I'm from the UK. I grew up in Hull. I got all of my education here, and then I moved to the US. And I'm now at Iowa State University. Here's a pretty picture of our campus. It's a beautiful campus, lots of trees, lots of plants. But some of you may not be familiar with US geography. And so here is America. Here's New York, California. This red state here is Iowa. So we're right in the middle of the country. And if we look at agricultural production, this is the market value of crops just a few years ago throughout the US. You see this big green patch right here? This centers around Iowa. So we're one of the major producers of agricultural products in the country. This means that plant biology is really important to Iowa. And in particular, there's a lot of interest in research on crop plants. The major crops that we produce are corn, maize, and soybean. And we grow these because we have really good soil. But we also have a lot of environmental challenges, particularly the climate. And so there's a lot of interest in trying to understand how plants, including crops, can tolerate these conditions, and also how to improve crops so that they can better tolerate these environmental stresses. And so it's a really good place to study plants' environmental interactions. And just to show you what I mean, this is what Iowa looks like. These are Iowa fields. Obviously, this is in the winter. You can see there's farms dotted around. Most of it is agricultural land. This is maize growing in a field just outside Ames, where Iowa State University is. This is a soybean field being harvested. Another maize field. So if you look at the Iowa scenery, it's mostly plants. We grow plants everywhere. Few towns, lots of agriculture. And so this is really why it's such a good place to study this, because people are interested in plants and people want to know about plant research and how we can help the agricultural economy in terms of improving plants. And just to show you some Iowa plants close up, these are a couple of people from my lab. We do a little bit of work with maize. These are maize plants, and this is during pollination time. This is one of our research fields, where these are a PhD student and a postdoc who are pollinating maize. But saying that, most of the research in my lab is done with the model plant Arabidopsis. So this is much easier to work with than maize. So most of the time, we're using Arabidopsis to give us information that we can then translate into crops like maize to try and understand better how to help agriculture. And so actually all of the work that I'll show you today, we've done with Arabidopsis. And we're now trying to do some of the same things with maize. And so when we think about agriculture and environmental stress and how we can improve plants, there are a few different things that we need to think about. But of course, when it comes to agricultural productivity, the important thing really is the yield. How much product do we get from these crops? And what that product is depends on the crop. But in Iowa, we're looking at beans and corn. And this is a map, obviously, showing the potential for closing the global yield gaps. So the yield gap is the difference between the potential yield that a crop can give and the actual yield that you get in the field. And these are different because of usually growth conditions that are not optimal, attack by pathogens, various things that reduce the yield of a plant. So the aim is to raise that actual yield that you get so that it gets closer to the theoretical optimal yield that you could get from a particular piece of land. And so this is a map showing the yield gaps in different places in the world. Here's Iowa right here. You can see there's actually a big potential for improving crops in Iowa. But this is also important in many other places, including Europe and Africa, Asia. Most places in the world are not getting the maximum yield from their crops that they could. And there are various ways that you could potentially improve the yield from crops. Big one is improving the nutrient supply. Improving water supply, certainly in some parts of the world, is critical. And then, of course, improving the crops themselves. And so in my lab, we're really interested in all of these aspects and how we can improve plant productivity and just plant growth during these different environmental conditions. And we do this by studying a very specific process. And that's a process called autophagy. And if you look at the word literally, auto means self and phagy means eating. So this is self-eating. In the context of a plant, or actually any other eukaryotic organism, this means the digestion of a cell's components by that cell itself. And I'll show you that in a minute, what that means. So this is really what I'm going to talk to you about, this self-eating or autophagy process. And I just wanted to remind you what a plant cell looks like. I know you've all been looking this week under microscopes and should be fairly familiar with cell structure. Here's a very simplified model. And remember, in a mature plant cell, usually the biggest organelle is the vacuole. And that's really the organelle that's going to be highlighted today. The vacuole is the organelle that does this autophagy process. But of course, this vacuole is surrounded by lots of other organelles, mitochondria, chloroplasts, the nucleus, the ER. And these other organelles are the components that can be digested by autophagy. So we have the vacuole that does the digestion. All of these other organelles potentially can be digested. And so if we look at a real plant vacuole, this kind of image might be familiar, at least to some of you, that have looked, that have done the cell biology practical already this week. This is a picture of an Arabidopsis plant. These are stem cells, that is cells of the stem, not stem cells as in meristematic cells. And this is a transgenic plant. So this is expressing a GFP fusion, a fluorescent protein fusion, with a protein that localizes to the vacuolar membrane. So everywhere that you see green fluorescence, you're looking at a plant cell vacuole. Most of these cells, they're fully expanded, they're mature cells. They have just one vacuole in each of them. So basically, each time you see a green fluorescent vacuole like this, you're looking at a cell. So in between these two vacuoles is the cytoplasm, the plasma membrane, and the cell wall of these cells. So hopefully you can see from this that most of the cell is vacuole. It takes up nearly the whole cell volume in these mature cells. And it has lots of different functions. So the vacuole is important for turgor. And so this turgor pressure gives the plant structure. The vacuole takes in water, and that's one of the things that helps a plant to maintain its structure. And you can tell this because if you forget to water your plants, they start to wilt. And this is because the vacuoles have lost their turgor pressure and are no longer pressing outwards against the cell wall to keep the plant upright. They're important for homeostasis, or maintaining a balance of different molecules in the cell. They're important for storage, especially in things like seeds. Vacuoles can store proteins that can then be used by a seed when it germinates. And then finally, the one that I'm really interested in is degradation, or breakdown of other molecules. The vacuole is full of enzymes that can degrade proteins and nucleic acids and polysaccharides. Pretty much any macromolecule that you can think of can be digested and broken down by the vacuole. And so that's the process that I'm interested in. How does this degradation, this digestion process occur? And also, if you think about it, how do molecules get into the vacuole to be digested? That's really where this process of autophagy comes in. And so here, if you imagine that the entire background to this slide is a cell, a single cell in a plant. Here's the vacuole over on this side. And here are a bunch of things that are going to be digested by autophagy. And so you can see here in orange is a mitochondrion, and purple is a ribosome, and these are, who knows what, some molecules that are present in the cytoplasm. And this red line here indicates a membrane. And so when this autophagy process is activated, this cup-shaped membrane starts to form. And it begins to surround whatever is going to be digested. And these membranes expand and eventually close up to give this structure that's called an autophagosome. And this is important because this is how we measure autophagy in plants, is by counting the number of autophagosomes that we see. So this autophagosome has got two membranes, and that's just because of the way that it forms. So it's got an outer membrane and an inner membrane. And if you can think of this in 3D, these are basically spheres. So they're completely enclosing this cargo. So you've got two membranes, and then in the middle is the content that is going to be digested. This autophagosome then moves to the vacuole, and the outer membrane of the autophagosome fuses with a tonoplast, which is the vacuolar membrane. This means that the inner membrane and whatever's inside the inner membrane ends up inside the vacuole. Because of all these enzymes that are present in the vacuole, that inner membrane and the contents then get digested. And so proteins are broken down back to amino acids, for example. Those amino acids can then be transported back out of the vacuole, and they can be used again by the cell to make proteins. And the process can be either selective or non-selective. And by that we mean that either an autophagosome can contain random contents, or the autophagosome can select specific cargo, for example, just mitochondria, to be digested. And these two processes, whether it's selective or non-selective, depends on the environmental conditions and the processes that the cell is undergoing at the time. I did want to show you an example of a movie. Hopefully this will play. Let's see. This is just showing you, it's actually a kind of a snapshot from a video game or a computer game that we've developed that I thought some of you might be interested in. And this is showing the process of autophagy. This is an autophagosome forming around a damaged chloroplast. The outer membrane of the autophagosome is fusing with the vacuole, which is the purple thing. And then the inner membrane and the chloroplast inside it end up inside the vacuole. And then they get digested into small molecules, and those small molecules can be reused. This is a game that we've developed to help mostly high school students, but also beginning undergraduates to learn cell biology. If any of you are interested, the website for the game is on the top. So we've just incorporated the process of autophagy into the game, but it's much bigger than this. You enter a plant cell and can fly around in a bioship and investigate the function of different organelles and look at what happens under different conditions, such as high light conditions. And so this process of autophagy is important for a lot of different reasons. And I wanted to start out by giving you an example, probably of the best studied situation in which autophagy is important. And that's nutrient stress. Showing you here some pictures of plants, corn and rice plants, that are in conditions of nitrogen deficiency, so they don't have enough nitrogen. And remember, nitrogen is important because it's incorporated into a lot of different molecules, including things like proteins and nucleic acids, DNA, RNA. And so plants need a lot of nitrogen. It's a macronutrient, and it's critical that they get enough. And if plants don't get enough nitrogen, they start to yellow. You can see the symptoms of yellowing here on these corn leaves. And then they also have stunted growth. And so it's very detrimental to plant growth. And so if you think about how plants get nitrogen, plants, of course, photosynthesize to fix carbon, but they have to take up minerals, including nitrogen, from the soil. And so if there isn't enough nitrogen or other minerals in the soil, then they're going to have problems in growth. And this can be a big problem agriculturally. But it's fairly simple to solve this problem. You just add fertilizer that contains things like nitrogen. Okay, so then if it's so simple, what's the problem? The problem with this is that fertilizer takes a lot of energy to produce, so it's not particularly good for the environment, and it's a limiting resource. There's a limited amount of fertilizer that we can generate. And so at some point, we may run out or stores may be depleted. And about half of the world's food supply is dependent on this inorganic fertilizer. So just because we can add fertilizer and the plants will grow nicely again, doesn't mean that this is always going to be the case. So we'd like to look for alternative approaches to help a plant retain as much nitrogen as possible and grow potentially under lower nitrogen conditions. The other problem is an environmental one, which happens after you apply the fertilizer. And I'm going to use Iowa as an example again here. Here's Iowa. This eastern border of the state is the Mississippi River. And if you look at the Mississippi River in Iowa, it's this big, wide, beautiful river. Lots of trees growing on it. But the Mississippi goes all the way from Minnesota down to the Gulf of Mexico and comes out right here in Louisiana. And I'll show you a blow-up of just this bottom part here. So here is where the Mississippi comes out. And this coloring here is showing you the dead zone. This is where basically the ocean is dead, nothing much can grow there. And that's because of pollution from nitrogen fertilizer. That nitrogen enters at places like Iowa, runs down the Mississippi, and then kills off all the fish in the Gulf of Mexico. So it's actually a huge environmental problem that farmers are applying a lot of fertilizer to their crops. Some of it is used by the plants, some of it is running off into the rivers and polluting them. So another reason why we might want to reduce the use of fertilizer. So one possibility for helping plants to get by with less nitrogen so we don't have to add as much fertilizer is the idea of autophagy, which is actually a nutrient recycling process. And autophagy has, this is a major function of autophagy that we know about, but it actually has a few others. So remember, autophagy degrades macromolecules within cells and it degrades organelles. So if you take a protein, you break it down back to amino acids. Those amino acids can then be reused by the cell rather than the plant having to make new amino acids which would require nitrogen. So it's a way that the plant can recycle its components rather than needing new nutrients taken up from the soil. Autophagy is actually under normal growth conditions. If you have plants growing in a nice, healthy environment, it's not particularly important for them. And so here, what I'm showing you on this slide are two sets of plants. One, this is wild-type plants. These are just normal plants growing in soil. The other plants on the right-hand side are plants that are defective in autophagy. So these are mutants that have been blocked in the autophagy pathway, so they can no longer do it. And under normal conditions, they look pretty similar to wild-type plants. But autophagy is activated when the plants are exposed to environmental stresses. And so under conditions like the addition of salt or drought stress, you can see that our plants that are defective in autophagy are not nearly as healthy as the wild-type plants. So this tells us that autophagy is important for the plants to respond to environmental stresses. And autophagy is activated under a pretty wide range of environmental stresses and helps the plants to tolerate under those conditions. The first one is starvation. And I've already talked a little bit about that. This is really the one that's best studied. It's important for nutrient recycling. But autophagy is also activated and important in a lot of other conditions. And in most of these conditions, it's thought that rather than being important for nutrient recycling, it's actually important for damage control. So when you have something like drought, proteins and other molecules within a plant cell get damaged. And if those damaged proteins accumulate, they can be really toxic to the cell. And so the cell will digest these using autophagy so that it can prevent the buildup of these toxic molecules. It's also activated in response to pathogens and then some environmental conditions, including senescence or aging of the plant. When a plant gets old and it's going to produce seeds, usually the leaves will turn yellow and the molecules within those leaves will be recycled to the seeds so that they can produce proteins. Also important during programmed cell death, and this is a developmental process. And it's a little bit of a conundrum in that autophagy, as we study it in response to environmental stresses, helps the cell to survive. But if autophagy keeps going and digests the entire cell contents, then it will kill the cell. And so we always have this balance between cell survival and cell death if autophagy continues for a long time. And so it has to be very tightly controlled so that you don't end up killing the cells that are trying to survive. And just to give you some examples of these other kinds of stresses that we're interested in, for example, salt stress, you might think that plants only encounter salt conditions when they're by the ocean. But actually, salt stress, it can be quite a big problem in agricultural settings where plants have been irrigated for a long time and where the irrigation has been maybe mismanaged or too much water has been used. Because what happens is the plants are irrigated, the water evaporates if the temperatures are high, and they leave behind the salts that were dissolved in the water. And so you can end up with situations like I'm showing here. This is a field in California, and this white stuff here is actually salt, where this field has been watered so much that the salt has been left behind, and now nothing much is going to grow in this field. So it can be a big problem for agriculture in that sense. Another big one is temperature, and I'm going to come back to this in a little while. Temperature, if we look, this is Des Moines, so this is close to Iowa State University. We have huge differences in temperature throughout the year, and also between the high temperature and the low temperature. So plants have to be able to respond to high temperatures and to low temperatures and to survive these conditions. And so some of the major questions that we're asking in my lab are simple ones like, when is autophagy activated, under what conditions? And then to get it a little bit more complicated, what are the functions and the substrates of autophagy under these conditions? And then also, how is autophagy regulated? How is it controlled? We know that it's not very active under normal conditions and that it's activated during stress. And this last question is important because if we think about autophagy, you might think to improve stress tolerance, you could just switch on autophagy all the time. But that's actually not a very good strategy. And the reason is that plants have a balance between growth and stress responses. And so very often, if you activate stress responses, the plant will switch off growth. So if we activate autophagy permanently, there's a good chance that growth is then going to decrease, which is the opposite for agricultural plants, the opposite of what we want. And so instead, we still want autophagy to be activated by stress conditions, but maybe it can be activated more strongly or more quickly than it would otherwise be, and this can help a plant to tolerate the stresses. And so in order to be able to do something like that, we really need to understand how autophagy is controlled, how is it activated. One example of how autophagy is activated, one pathway, is this one that I'm showing here, which is called the target of rapamycin complex. This is a protein complex. And it was originally identified in animal cells, because animal cells also have an autophagy pathway that's actually really important in things like disease processes in cancer and in neurodegenerative diseases like Parkinson's or Alzheimer's. And so it's been quite widely studied for that reason. And we found in plants, in Arabidopsis, that they contain this protein complex similar to the one in animal cells. It has 3 different polypeptides. TOR itself, this is a catalytic subunit, and TOR is a protein kinase, which means that it adds a phosphate group to other proteins, and that controls their activity. So the TOR complex really controls the activity of other proteins. And then it has 2 other subunits, LST8 and Raptor, which help this protein kinase to function. And so when there are plenty of nutrients, TOR is active, and that activates growth. And at the same time, it represses autophagy. So this means that TOR is a negative regulator of autophagy. When TOR is activated by nutrients, autophagy is shut down because it's not needed. Now, I've simplified the TOR complex here to show you more as a cartoon. Now, what happens during nutrient deficiency is that the TOR complex is no longer activated. And this means that you can no longer get growth, and instead, you get activation of autophagy. So low amount of TOR activity means autophagy is active. But other stresses, as I just said, can also activate autophagy, things like drought and salt. So the question then is, do these other stresses also activate autophagy through TOR, or is it by a completely separate pathway? And this is one of the first questions that we wanted to answer. And to remind you, autophagy in plants occurs by this pathway, and these autophagosomes are the transport vesicles that move material into the vacuole for digestion. And we can count the number of autophagosomes within a cell, and this will tell us how much autophagy activity there is. And to do this, so that we can see the autophagosomes, we label them with a fluorescent protein or a fluorescence tagged protein that's found on the autophagosome membrane. So this means that we can see these autophagosomes in cells when autophagy is activated. And this is what it looks like. So this is microscopy of a plant cell. And if you look over here, this is our confocal microscopy. And these little bright dots that you can see inside these cells are the autophagosomes that we've labeled with a fluorescent marker. And so the number of these that we can count gives us a measure of autophagy activity. And here, I'm just showing you that this TOR complex is a negative regulator of autophagy in Arabidopsis. AZD is a chemical that can inhibit TOR. And so we can add this to the plants, and you can see that in wild-type plants, under normal conditions, we have a very, very few autophagosomes, so a very low level of autophagy. If we add this AZD TOR inhibitor, you get a huge increase in the number of autophagosomes that we can detect. So this tells us that TOR is a negative regulator. We can also look at genetically at this. So Raptor, if you remember, is one of those other components of the TOR complex. And so we can make mutants in the Raptor and see what effect that has on autophagy. And there are 2 genes for Raptor in Arabidopsis. One of them doesn't have too much effect, but the second one, this Raptor 1b, again, if you knock it out, this is a mutant, you now get a huge increase in autophagy. And so again, this means that TOR, the TOR complex, is a negative regulator of autophagy. So if you have high TOR activity, you have low autophagy activity, and vice versa. And we can use different assays to look at this. That previous one was in roots. We can also use a transient assay where we express our autophagosome marker in leaf protoplasts. So we digest the cell wall from leaves and release the individual cells. And then we can express any markers that we want in these cells. And we get the same results. This is showing you the Raptor mutants again. And the Raptor mutants have autophagy even under normal conditions when usually autophagy would be shut down because of TOR activity. To investigate the conditions in which TOR is important for autophagy, we use transgenic plants that have an increased amount of TOR in them. So these are TOR overexpressing lines. They have increased TOR activity, and so the prediction would be that if you have an increase in TOR activity, you would have a decrease in autophagy. So we looked into different conditions that would normally activate autophagy to see whether we did indeed have a decrease in autophagy activity in these lines. And we counted the number of autophagosomes again, and we looked into different conditions. So in this graph, we're showing you, here's just normal conditions, wild-type plants. And the control is in blue. And then if we starve these of sucrose, so this is a carbon deficiency, we get an increase in autophagy in red. If we starve them of nitrogen, also induces autophagy, we get an increase in the number of autophagosomes in green. If we look in our transgenic lines that have increased TOR activity, what we see is that actually under sucrose and nitrogen starvation conditions, we have decreased numbers of autophagosomes, so we have a decrease in autophagy. So this matches our prediction. Now, TOR is known to be involved in animals in nutrient deficiency, but it's not known to be involved in other stress responses. So our next question was, if we look at other conditions, is TOR also involved in activating autophagy? And so we looked under salt stress, which is here in red. Again, salt stress activates autophagy in wild-type plants. And we looked under mannitol produces osmotic stress, which mimics drought. So here we're looking at, in the lab, at the equivalent of drought in the field. Again, activates autophagy in wild-type plants. And again, in all of our lines with increased TOR, we see that the autophagy is repressed. It's no longer activated under these conditions. So this was a little bit surprising because TOR was not known to be involved in these conditions, but it seems that TOR is also involved in autophagy under these other stress conditions in addition to nutrient stress. And we could do the same thing looking at our leaf protoplasts, and again, under both starvation and salt and drought conditions, we see that in our lines that autophagy is decreased if we overexpress TOR. But this doesn't happen with all stresses. It happens with starvation and salt and drought. But if we look under other kinds of stress conditions, and here we're looking at oxidative stress, which we're just adding hydrogen peroxide, and this mimics the kinds of things that happen, for example, when plants are exposed to excess light. And then under ER stress, and this mimics, these are chemicals that can mimic the unfolding of proteins that happens during heat stress, for example, and also some other stress conditions. So because we're doing these in the lab, it's easier to use these chemicals to mimic the stresses than to use the actual stresses themselves. And what we find is that if we look, for example, oxidative stress, wild-type autophagy is induced as we expect, But in all of our TOR overexpressing lines, we still get autophagy. So TOR overexpression doesn't repress autophagy under oxidative stress conditions. And the same thing using our ER stress, our unfolded protein conditions. And so it seems that autophagy is induced by oxidative stress and ER stress, but it's TOR independent. TOR has no effect on activation of autophagy under these particular conditions. And we can, again, confirm this using our protoplast assays. So we use two different methods in two different tissues, leaves and roots, and see the same thing. And so basically what we see is this TOR complex, which activates growth and represses autophagy. And it's controlled by nutrient stress, and it's controlled by salt and osmotic stress. But autophagy is activated by oxidative stress and ER stress independently of the TOR complex. So we have at least two different pathways for the control of autophagy. One of them requires TOR, and one of them does not. So the situation is getting a little bit complicated. And so our next question was, so then how is autophagy activated under these different conditions? And I'm just briefly going to tell you about some work that we've done on ER stress and how autophagy is activated. So ER stress, I mentioned, is accumulation of unfolded proteins in the endoplasmic reticulum. And so this happens because protein folding needs exceed the capacity of the machinery, but basically it's usually caused by heat stress in the field. It gets too hot, proteins can't fold properly, and so they accumulate. And because the ER is a major site of protein synthesis, this is one of the main places that they accumulate. And so you get these unfolded and misfolded proteins, and this causes stress on the cell. And we can mimic this using chemicals as DTT and tunicamycin. And so I've really already shown you that autophagosomes and autophagy is activated during ER stress, and autophagosomes accumulate. And we actually know what the function of this is. It delivers ER to the vacuole, and the ER is actually degraded by autophagy during ER stress. So here we've got an ER marker in green and an autophagosome marker in purple. And if you look at the arrows, these two co-localize, at least partly. So this tells us that ER is being delivered in autophagosomes. And we've done some other work to show that it ends up inside the vacuole and is degraded. And this is important because we have mutants that can no longer do autophagy, and they're really sensitive to ER stress. So if you look at survival of plants on these chemicals that cause ER stress, then autophagy mutants shown here, these are just blocked in the autophagy pathway, have much lower survival than wild-type plants. So autophagy is important for survival of ER stress, and also heat stress. Same kind of idea, we'll look at survival after heat stress. Autophagy mutants survive more poorly than the wild-type plants. So it's important for tolerating heat stress. ER stress signaling is quite well studied, but not in relation to autophagy. It's in relation to the upregulation of proteins that help in protein folding. And you don't really need to know the details of this, but just note that this ER stress signaling is initiated by unfolded proteins in the ER. And so we looked to see whether that was important for autophagy as well. And we used a chemical chaperone called PBA. And a chaperone is something that helps other proteins to fold. So if we add this chaperone, the amount of unfolded proteins should decrease in the cell. And so we added this chaperone and then looked to see the effect on autophagy. And what we found, so here is an ER stress where we've activated autophagy, lots of autophagosomes. If we add this chemical chaperone to it, it brings the number of autophagosomes way down again. So this means that unfolded proteins were important for the activation of autophagy under these conditions. It's not important under all conditions, because if we starve them for sucrose, then this PBA has no effect on activation of autophagy. So it's specific to these conditions that cause accumulation of unfolded proteins. We can see the same thing during heat stress, that PBA, heat stress induces autophagy compared with the control. PBA reduces the number of autophagosomes again. So again, it's heat stress and these chemicals are kind of doing the same thing. And then we know that one of the proteins that's important for ER stress signaling is a protein called IRE1. This was already known to be important for the activation of ER stress, but we didn't know anything about its role in autophagy. And so we looked at mutants that are blocked in this particular factor and looked to see what effect they have on autophagy induced by ER stress. And I think it's easier to see probably on a graph. So here is a control, no autophagy. We add these ER stress agents, we get activation of autophagy. But in an IRE1 mutant, which is the gray bars, we no longer can get activation of autophagy during ER stress. And this again is specific just to this ER stress, because if we look during sucrose starvation, the IRE1 mutant can still activate autophagy. So this is telling us again that we have multiple signaling pathways, different ways of activating autophagy, depending on the environmental stress that we impose on the plant. And also during heat stress, IRE1 is important because the IRE1 mutant can no longer properly activate autophagy during heat stress, again indicating that the heat stress and these chemicals are acting in the same way. And so if we go back to our pathway for how autophagy is controlled, you can see that we have at least two different ways to activate autophagy. And this is where it gets complicated when we're thinking about stresses in the field, because if you imagine in an agricultural setting, plants can be under multiple different stresses at the same time. They can be under heat stress and drought and nutrient deficiency all at the same time. And yet different pathways activate autophagy under these different conditions. So we have to keep that in mind when we're trying to design strategies to improve plant tolerance to these stresses. So we have these two pathways. One centers around this TOR complex, and that nutrient deficiency and salt and osmotic stress or drought feed into this pathway to control both growth and autophagy. ER stress and oxidative stress are independent of TOR, but at least ER stress activates autophagy through this other unfolded protein signaling pathway via IRE1B to activate autophagy. So two separate pathways. And so now in the lab, we're trying to understand more about these pathways, what other components are involved, and how can we manipulate them to more effectively activate autophagy when it's needed by the plant. And so finally, I just want to mention the people in my lab that have helped with this work. The ER Stress Project is actually a collaboration with another lab at Iowa State, actually, which is next door to mine. And they're experts on ER stress. And these are the PhD students and postdocs in my lab that have done different aspects of work on this project. So I will leave it there, and thank you for listening.